Good morning, students. Today we are going to start new chapter, chapter number 11, Biotechnology Principles and Processes. The chapter includes topics like Principles of Biotechnology, Tools of Recombinant DNA Technology, Processes of Recombinant DNA Technology. Biotechnology deals with the techniques of using live organisms or enzymes from organisms to produce products and processes that are useful to human. If we split the terms bio and technology, bio means life and technology. When the organisms are used with the help of the technology for the welfare of human being, the branch of science is known as biotechnology. In this sense, making curd, bread, or wine, which are all microbe mediated processes, could also be thought as a form of biotechnology. However, it is used in a restricted sense today to refer to such of those processes which use genetically modified organisms to achieve the same on a larger scale. Further, many other processes or techniques are also included under biotechnology. For example, in vitro fertilization leading to a test tube baby, synthesizing a gene, using it, developing a DNA vaccine, or correcting a defective gene are all a part of biotechnology. So today we have moved towards a wider area of biotechnology like test tube babies, gene developing DNA vaccines, etc. The European Federation of Biotechnology (EFB) has given a definition of biotechnology that encompasses both traditional view as well as modern view of the biotechnology. The definition given by EFB is as follows: the integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services is known as biotechnology. Principles of biotechnology. Among many, the two core techniques that enabled birth of modern biotechnology are genetic engineering. First, techniques to alter the chemistry of genetic material, either DNA or RNA, is referred to as genetic engineering. To introduce these into host organism and thus change the phenotype of the host organism. Second step is bioprocess engineering. Maintenance of sterile, that is microbial contamination free ambience in chemical engineering process to enable growth of only desired microbe or eukaryotic cell in large quantities for the manufacture of biotechnological products like antibiotics, vaccine, enzymes, etc. Let us now understand the conceptual development of the principles of genetic engineering. You probably appreciate the advantages of sexual reproduction over asexual reproduction. The former provides opportunities for variation. Former means sexual reproduction provides opportunities for variations and formulations of unique combination of genetic setup, some of which may be beneficial to the organism as well as the population. Asexual reproduction preserves the genetic information, while sexual reproduction permits variation. Traditional hybridization procedures used in plants and animal breeding very often lead to inclusion and multiplication of the undesirable genes along with the desired genes. The technique of the genetic engineering, which include creation of recombinant DNA, use of gene cloning and gene transfer, overcome this limitation and allow us to isolate and introduce only one or a set of desirable genes without introducing undesirable gene into the target organism. When the two DNA combine, then the molecule is recombinant DNA. Gene cloning means copying the gene, and gene transfer means transfer of gene from one organism to another. Do you know the likely fate of a piece of a DNA which is somehow transferred into an alien organism. Most likely, this piece of DNA would not be able to multiply itself in a progeny cell of an organism, but when it gets integrated into the genome of the recipient, it may multiply and may be inherited along with the host DNA. This is because the alien piece of DNA has become a part of chromosome which has the ability to replicate. In a chromosome, there is a specific DNA sequence called the origin of replication, ORI sequence, which is responsible for initiating the replication. Let me remind you what is replication. Copying DNA is known as replication. So, therefore, for the multiplication of any alien species of DNA, it needs to be the part of the chromosome which has a specific sequence known as origin of replication. Thus, an alien DNA is linked with the origin of replication so that this piece of DNA can replicate and multiply itself in the host organism. This can also be called as cloning or making multiple identical copies of any template DNA. Let us now focus on the first instance of the construction of an artificial recombinant DNA molecule. What is instance? First example. The construction of the first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid, its own plasmid, autonomously replicating circular extrachromosomal DNA is known as plasmid. Of Salmonella typhimurium, Stanley Cohen and Herbert Boyer accomplished this in 1972 by isolating the antibiotic resistance gene by cutting out a piece of DNA from the plasmid which was responsible for conferring antibiotic resistance. The cutting of DNA at the specific location became possible with the discovery of the so-called molecular scissors called restriction enzyme. Actually, these enzymes produce a cut to a specific sequence. They cut the nucleotides, they cut the nucleic acid, either DNA or RNA, at a specific nucleic acid sequence. The cut piece of DNA was then linked with this plasmid DNA. This plasmid DNA acts as a vector to transfer the piece of DNA attached to it. You probably know that mosquito act as an insect vector to transfer the malarial parasite into the human body. In the same way, a plasmid can be used as a vector to deliver an alien piece of DNA into the host organism. जिस तरह मॉस्किटो मलेरियल पैरासाइट को हमारी बॉडी में इंसर्ट करता है, the same way this plasmid vector inserts the gene into the cell. The linking of antibiotic resistance gene with the plasmid vector became possible with the enzyme DNA ligase, which act on cut DNA molecules and join their end. This makes a new combination of circular, autonomously replicating DNA created in vitro, that is in laboratory, and is known as recombinant DNA. When this DNA is transferred into Escherichia coli, a bacterium closely related to Salmonella, it could replicate using the new host DNA polymerase enzyme and make multiple copies of it. The ability to multiply copies of antibiotic resistant gene in E. coli was called cloning of antibiotic resistance gene in E. coli. Let us understand through this video. The DNA is first extracted from the bacteria and taken into the test tube. Restriction enzyme is used to cut the extracted DNA into the gene-sized pieces. In the another test tube, the extracted bacterial plasmids are cut using the same enzyme so that the same sequences are cut and same ends are produced, and when both are mixed, they will join with each other in the presence of enzyme DNA ligase. Some plasmids are joined themselves without the cut extracted DNA, and some have the cut extracted DNA. Those which have the extracted DNA are known as recombinant plasmids. Bacteria are then added to the test tube. The bacteria gain the plasmids by the electroporation. The plasmids inside the bacteria are of two types, some with the recombinant DNA and some with the original DNA. Those which have the recombinant will be antibiotic resistant and they will grow on the antibiotic medium, but the others which are not recombinant will not grow on the plate with the antibiotic. So those organisms which we obtain on the media containing antibiotic are recombinants, means they have took up the extracted DNA and the bacteria have been transformed for that antibiotic resistance. The genetic engineer finds the colony that has been transformed and transfer it to the another petri dish and allowed it to multiply and develop a large colony with the hundreds of copies of our gene of interest, because the bacteria was transformed, and when it divides, the gene of interest also divides.